Thank you very much, Dave. That was a brilliant talk, so thank you for introducing that. So next, I'm just going to give you a bit of an overview again, just like what Dave's done basically, but this time on biobanking. So we're going to mention this word biobanking quite a lot today. Biobanking to save breeds and species. So an outline of what I'm going to talk about. I'm going to introduce what is biobanking, why do we need to do it, the different materials that can be banked, some examples of people already doing it, and talk about which biobanking is right for you. Can I quickly just, can you hear me at the back? Because I sometimes, brilliant, thank you. I sometimes mumble and don't realise I'm doing it. So.
Excuse me, can I just ask, will we be able to get copies of these slides at any point? Or do we need to scribble?
You can scribble, but we can look at that for you if you'd like to do that.
Yeah.
We're also recording. We've got a little tripod going, so recording the whole day as well. And my aim is to be able to trim that whole recording up into sections, and we could potentially post out on the website Dave's talk if you just want to listen to Dave and this one. So yeah, but we'll look at notes for you.
Thank you.
So just a brief introduction to me. You already know who I am until I said it. So I'm manager of Gemini Genetics. We've got a trade stand out there. We are a genetic preservation company partnered with a cloning company. But I'm not here just to tell people to clone their animals today. A real key message I want to get across to you as a rare breed population is about genetic preservation. So banking skin samples from animals, so you've got a whole genetic set preserved for the future. So that's what I want to talk about today, really. I started off as a lab technician at Stallion AI, so I do know a bit about semen as well, and I help Dave occasionally with TCIs in particular, it seems. And I'm also a scientific advisor and lab technician for Nature's Safe. So that means I've got experience with rare breed animals and rare breed species. Really important person to introduce you to though as well, and she's not in the room because she's downstairs still registering people, is my lovely student Eloise Bates. I believe her family might be in the room. I've not met them to know who they are, but Eloise is brilliant. You will get to see her today. She's a bioveterinary science student and she's helped me a lot today, so we need to pay her a thanks in her absence because she's downstairs. So just to start off with, what is biobanking? So biobanking is a collection of biological and genetic material for future use. So as I say, it's going to be something that we're going to talk about a lot today. And it maybe doesn't sound the most interesting of topics, but it actually really is once you start delving into it. And it's really a concept that I want you to take home with you today. So I've got a quote up there from the Frozen Zoo. It's quite, we're going to talk about the Frozen Zoo a bit more later on. So we must collect things for reasons you don't yet understand. So we are future-proofing and providing future security against the unknown of what's coming up against us in the future. It's a concept that is across both human, animals and plants. So actually the most developed sector, and I'll talk about this later, is for plants. So the plant industry, they've already got seed vaults and things like that. So they've already got lines and species of plants backed down. The animal sector, though, we really need to start catching up in certain departments on that, in having a secure biobank for the future. But it's also something that they're looking at for humans as well, biobanking different materials for humans, for future research and so on. So biobanking, it's very much the buzzword of the century. We seem to be, as a company, across all the different hats that we wear, talking about biobanking almost every day sometimes. And this is just a publication I found. It stopped in 2019. It was published mid-2019, which is why the graph stopped there. But my prediction would be that it's possibly kept going up since then, because as I say, we keep talking about this word increasingly all the time. So that's why I wanted to introduce it to you today. So why do we need to biobank? It's basically protection against the future unknown. So we are, and I'm going to talk about later on the charity Nature's Safe, where I'll introduce things like climate change, growing human population, all the future unknown that we are unfortunately facing. You know, we are going through the 6th mass extinction at the minute. We are going through a period of climate change. So our future world is actually one that we're not quite sure what that's going to look like yet. So that's the real key thing, why we need to start banking samples now while we have them available to us. Because if we don't take action now, we will lose that material and we won't be able to recall it. So that's the one key thing there. Other reasons why we need to biobank are for breed and species genetic diversity. So there will be in existence each generation key individuals, or numerous ones of them, that are really key to that breed or species genetic diversity and future sustainability potentially. So this is why we need to be thinking about proactively banking them so that we've got them in the future to be able to recall them, whether that be to recall a specific trait that we've lost, or to introduce some resistance to disease or some future element that we need to provide the breed with some sustainability against. So this is really important while we bank them now, while we've got them. It may also enable us to go back and improve a breed. So we might find that we go down one line of breeding and we reach a certain goal or target, but then we actually think, oh, I wish I could introduce something that I've actually outbred. So that's another really important reason that we need to keep them. Recall a lost traits, and ultimately breed and species survival. So Dave was talking about we're breeding within a closed gene pool. So within that concept, it is therefore really important that we are banking material down, and we're banking a diversity of material down, because inevitably within a closed pool, if we don't have a high number of individuals to call back upon, we're going to lose diversity as we go through the generations. Protection against unexpected population challenges. So I've got a couple of slides in a minute, but we all know this is obviously a human example, but we've had COVID, we've got bird flu at the minute for avians. There's going to be disease challenges that come up in our future, and we really need to start protecting ourselves against that. I've got some figures in a minute just to show you an example of how devastating bird flu's been. But again, it really reiterates that we need to bank samples while we've got them, because once they've gone, they have gone, and we can't get them back. More on the human side of things, but they begin to think about it for animals too, is really developing personalised medicine. So if we can bank samples, we can begin to learn more in depth about genetics and how we can really personalise and tailor medicines and treatments to us, and therefore improve, if we're talking about humans, our healthcare and so on, but also our animals and their longevity and potentially some of the conditions that we need to work with within certain breeds, and understanding of health and disease. So going back to that original quote, the samples that we bank could hold information that we don't even realise we need in the present generation. So it's all about getting it back now so that we've got it for the future. So this is a plant example, but as I said, the plant industry are more advanced in terms of banking their seeds so that they've got them for the future. And this is one example of why they need to biobank. So this is an example in bananas, Panama disease. It's a current very severe threat facing the banana industry. Up to 80% of production is at risk from this condition, which there is no cure for at the minute. It's caused by a fungal disease. And I know we're just talking about bananas here, but there'll be lots of people that like bananas, and hopefully lots of people that won't want them to go extinct. They're also a staple food for certain countries. They will be a staple food for them. But this concept is transferable to other crops and other animal species, breeds, etc. So this is really a high point. You know, they've got material banked down, and this is why these disease challenges are why we need to start thinking about banking what we've currently got access to. And the other, so more animal related one is bird flu. So globally, the disease so far has killed 140 million poultry since October 2021, and that's really astronomical. And that's wild birds, bush agricultural birds. So we've lost biodiversity as well as affecting our food production. And of course, bird flu is still ongoing at the minute. So again, not directly relatable, you know, it's not a canine condition, but we could potentially in future encounter something that could challenge our breeds. And this is why we need to be banking down materials now. So we want to, when we talk about biobanking, we want to collect a diverse range of samples. So we don't just want to, semen fusion is great, but we're only banking males there, and we're only banking half their genetic set. So we need to bank multiple sample types because they're going to have different uses in the future, and also we don't want to be just reliant on one strand. That's our only lifeline, is one method of preserving an animal. So we can bank tissue samples, and I'll talk about that later on for you. Reproductive samples, so that can be the semen, but also the reproductive organs. So we can bank little bits of testes and ovaries with view of being able to wake them up in the future and get them to produce fresh sperm and eggs for us. Banking hair and body fluids, that's more for doing research purposes rather than bringing back an animal, so to speak. And the key thing is about it being organised and catalogued biobanking. So with my Nature Safe hat on, there are, which is the Wild and Endangered Animal Charity, So there are different organisations banking their material at the minute, but we don't necessarily all talk to one another. And to make the most efficient use of things, we all need to be talking and working towards the same end goal, and also have things organised. So once you start biobanking, you'll soon realise that your samples you soon start from one sample, and all of a sudden you've got hundreds of them in the bank. So it's really important that you're doing it in a really organised way, and future-proofing the way you're organising stuff as well. So another element that we might work on in the future is having our biobanking material linked to clinical records and lifestyle information. So we could have biobank samples that we've genetic tested and so on linked to veterinary records, and that could help further our understanding of a certain disease condition and health, and overall improve health and longevity of a population. So some existing biobanking examples. So these are predominantly seed ones first. So hopefully you all heard of the Millennium Seed Bank, or the majority of you. Yeah, brilliant. I'd really like to go there myself one day. It's on the list. So it's the largest plant conservation program in the world, and it's really an invaluable collection of crops and plants for research and future food security. So they're banking down seeds so that we've got them stored there for if we have a disease outbreak in crops. There's over 1,000 seed banks across the world, and the number of seed banks are increasing all the time. That's our donkey. Yes, you'll get to see him later. He sounds enormous, but he's actually really tiny. Yeah, you can see him later. No, he thinks he's a big boy, and he's very stubborn at times as well. So yeah, for plants, basically, they've already been doing this and thinking about it, and our plants are researched before. Animal biobanks are much fewer. They're currently only present for wild and endangered animals, and even then, we're still kind of in the infancy for them. A key thing that we need to think about is banking samples that we can actually bring back to life again. So we can bank samples just to look at their genetic structure and so on, but a key thing if we want to sustain a population and protect against the future unknown is actually being able to bring back a living organism from the material that we're banking. And as I said before, for the animal biobanks at the minute, there's a bit of disconnect. Not everybody's talking to one another, so that's what we need to work on for the wild and endangered animals, getting people talking to each other about it more. So San Diego Zoo, as well as Nature Safe, that I'm going to talk to you about a lot this afternoon. San Diego Zoo is the largest and most diverse collection of exotic animal and reproductive material in the world. They've got over 10,000 living cell lines banked. So these guys, we started off with a quote from these guys. Basically, in the 1980s, these people were already thinking about banking samples for what they didn't know they were going to use them for in the future. And I've got some case studies coming up later where we've actually used, not myself, but the company we're partnered with, we've used samples that this zoo banked 30 to 40 years ago. In the present generation, we've used them to bring back animals to help with breed sustainability. So they wouldn't have known what they were going to use those samples for back then, but they banked them, so they've got them. And that's the key message that I want to relate to you, is that we need to, so we've got them there for the future. So for domestic animals, so your dogs and farm animals, there isn't yet an organised, defined system of biobanking for them. So hopefully that might be something we can now today. So what do we need to bank? I said we need a variability in samples. We don't just want to be reliant on semen to bring back a breed or skin samples alone. We need to bank different material. So we've got different lines to call upon in the future. So we've got skin samples, the reproductive organs, and the semen, and maybe also we can look at things like embryo freezing and so on, like things like that. We need a nice diversity, so we're not just balanced on one technology. So this is the diagram that I brought in from presentations I do for Nature Safe, but I've switched the wild animals to dogs. So in terms of ovaries, we can look at freezing eggs. The eggs are really difficult to freeze at the minute, but you know, it's a thing that we're looking at being able to do in the future. Eggs have got a really thick membrane around them and lots of fluid inside them, that makes them a lot more trickier to freeze than your sperm cell. But what we can do at the minute is preserve strips of the ovaries with potential for being able to regenerate them in the future and ask them to produce fresh eggs. That's obviously a post-mortem scenario. Then we've got testes, so we've got semen, and semen can be harvested both by just a regular collection from your dog, but we also work on post-castration epididymal semen extraction. So if the dog's castrated, we can have a go at removing the semen from the testes following castration. Can be variable in those results, and it's trickier for dogs than, so we do a lot of them for horses, and horses are generally quite easy. They've got nice big testicles. We can generally harvest the semen nicely from them. Dogs are a bit trickier, they're smaller, something that needs more work on it, but it is that option out there that we can try. And then the same thing with them as preserving strips of the testicular body as well to be able to wake that back up. And then the other arm of things is the skin samples. So one outlet of use of the skin samples is for cloning, and cloning does have an application for bringing back genetic diversity into a closed population, and I'll talk about that more in a moment. But the other application of it is to generate induced pluripotent stem cells. So basically to take a skin sample and turn it into, and this is a futuristic concept, but turn it into a sperm or an egg cell. So that is one that we are looking at as well, because that would enable us to generate new individuals with a fresh genetic set from a skin sample that's really easy to take, generally really successfully taken from all, whereas semen and other samples can be variable in quality. Generally, a skin sample, if you get it to us within the required time, it's going to regenerate the way we need it to. So that would be a brilliant technology if we can develop that in the future, and there are people working on that. So when do we need to biobank? So the quality of the biobank is only as good as the quality of the material entering. So we need to ideally biobank when a population is genetically diverse, so we can capture the different lines that we need potentially to recall upon for the future. And so when we're breeding within a closed population, I normally put this diagram up because you are essentially breeding within almost a funnel shape. So we want to be banking when the breed base is ideally quite wide, and we can then go back to individuals that have previously existed to bring back their diversity. And I'll show you more. This is the concept that they applied for the zoo animals that I'm going to talk about later on. They've cloned a black-footed ferret and a Polski's horse, and this is the concept that they used for them, basically. We want to biobank ahead of reaching the extinction vortex. So Andy's going to talk about the extinction vortex in greater detail, but once we've started going down this stage, once we start going into an irreversible population loss, that is really tricky to get out of. So we want to really have the foresight to be able to bank now while populations are more diverse, so that we've got the tools that we need to help stop ourselves going into this vortex in the future. I just wanted to give you a bit of context on the equine parallel that we work with. So our biobanking really started from our work with rare breed horses. And we can see, I've got a table over there with some of the average inbreeding for some of the horses. And so this is really, although they're equines, there'll be concepts that we can relate across to. So we've already began working, and we're working with them continuously to help bring back and bring a more sustainable future for these breeds. But I just wanted to show you a video, I'm hoping it's going to play, for the, it's from the Rare Breeds Survival Trust. So I borrowed that from their campaign. I always get little goosebumps whenever I hear that, watch that video. I think it's a combination of the music and the hard-hitting facts. So that graph was very much comparable to Dave's bloodhound graph almost. And you know, the figure that they put up there in, I think it was 2027 that the date was, that they could be extinct. You know, that will, I'm sure that will ring parallel to a lot of you guys potentially in the room with some of your breeds. So yeah, this is why we need to be acting now. Big words in there, and you'll see that we need to act now come up a couple of times potentially during the day. And that's a really hard-hitting video of where we are for horses, but also it will be comparable to a lot of you, your breeds today. So I'm going to talk about nature safe a lot in my second presentation. But for wild and endangered animals, Tullis mentioned it before, we are in the 6th mass extinction, largest predicted loss of biodiversity for 65 million years, up to 1 million species at risk of extinction. And it's highly relevant to us because it will change the world that we live in today. Our world is what it looks like because of all the ecosystems and the planet and climate relations that go on. So losing our species combined with our climate change will have a dramatic impact on our world. But also the fact that these species are going through extinction, it can be relatable to what some breeds are seeing on the domestic side. So extinction on the natural, on the wild and endangered side of things, it is a natural event that occurs. Not all species will be suitable for current climates and so on, so they will naturally trickle away. Some of them will become extinct, but you can see that the current rate is dramatically, significantly flying out of the water compared to what it should be. And that is the huge challenge and huge worry that we've got against us. And this is why we're working within the wild and endangered animal sphere to try and get samples back now, so that we can try and preserve as much as we've got available to us now as we're going down this rapid decline in species. So that is what I'm going to talk about in my second presentation as part of it. Nature safe, a living biobank, and the living concept is the real key thing here. We want to be banking samples that we can bring back to life and bring animals back to life with, so that we've got them for the future. And that is the end of my presentation.